Welcome, everyone. Thanks for tuning in to our May webinar. I'm Emily Whitmore. I'm with the Maine Aquaculture Innovation Center, and I'm really excited to announce today's webinar. We have an amazing team of researchers from the Aquaculture Research Institute at the University of Maine, and they're going to be presenting their work on finfish aquaculture in Maine related to innovations in health, breeding, and sustainable feed. Just a couple of housekeeping items before we start. One, we have a really exciting event coming up at the Down East Institute called Emerging Opportunities in Shellfish Aquaculture. The event's on June 8th, and Annie is going to throw a PDF in the chat with more information about that. Second, just a reminder to any businesses we have listening in, we have a variety of opportunities that are available for aquaculture businesses for innovation, scaling, and community outreach. And so check out the chat. There's going to be a link in there that brings you to our webpage for those resources. And then three, all of our webinars are recorded and available on our YouTube channel. So feel free to find our YouTube channel and check those out. I'm going to let everyone introduce themselves. I'm going to turn it over to Debbie Bouchard. One last thing, if you have questions, we're going to save them to the end of the webinar, but feel free to use the Q&A function at the bottom of your screen and pop your questions in, and we'll get them fielded at the end. All right, take it away, Debbie.
Thank you. Well, hello, everyone. As said, I'm Debbie Bouchard, the director of the Aquaculture Research Institute. I want to start by thanking the Maine Aquaculture Innovation Center for inviting us to share our work today. We're excited to talk about our research in finfish aquaculture, work that's shaping the future of sustainable seafood production here in Maine and beyond. I'll kick things off with a quick overview of the Aquaculture Research Institute, and then I'll hand it over to our faculty researchers who will dive into the projects they're leading. Next slide, please. So what does the Aquaculture Research Institute do? Our institute is a trusted source for science-based aquaculture research. Our mission is to support sustainable aquaculture in Maine and beyond, economically, environmentally, and socially. We do this by working across disciplines and partnering with industry leaders, tribal nations, government agencies, nonprofits, and other universities. Our research focuses on real-world challenges like improving feed sustainability, using resources more efficiently, and reducing environmental impacts to help shape the future of aquaculture. Next slide, please. Our work is guided by 4 strategic areas that together support a thriving, sustainable aquaculture industry. Healthy species. We focus on the health and nutrition of individual aquatic animals, ensuring they grow strong, stay healthy, and thrive in farm environments. Healthy ecosystems. We study how aquaculture interacts with the environment, from climate resilience to ecosystem modeling, to help farms operate sustainably and productively. Healthy populations. We look at the bigger picture like genetics, reproduction, and how species respond to climate change, aimed at supporting the long-term success of farm populations. Healthy communities. Aquaculture doesn't exist in a vacuum. We explore how it fits into society through research on food safety, policy, public perception, and community well-being. Together, these focus areas help build an aquaculture industry that is productive, resilient, and deeply connected to the needs of the people and the planet. Next slide, please. Our institute supports research and innovation across 5 key facilities throughout Maine, each playing a unique role in strengthening the aquaculture industry. I'll highlight 2 of them that are especially central to our current efforts in finfish research. First, the Aquatic Animal Health Laboratory, located within UMaine's Cooperative Extension Diagnostic Research Lab in Orono, is a vital part of our operations. This lab not only supports aquaculture producers through health diagnostics and research, but also functions as a contract research organization offering critical services to industry, academic, and government partners alike. Dr. Sarah Turner will provide more detail on this unique facility later in the webinar. Looking ahead, we're thrilled to soon break ground on the Sustainable Aquaculture Workforce Innovation Center, with construction set to begin later this month, right here on the Orono campus. Next slide, please. This new facility will serve as an aquaculture hub. It is a vital addition to UMaine's aquaculture infrastructure, designed specifically to support hands-on research, workforce training, and industry innovation. Focused on recirculating aquaculture systems, the facility will provide a unique environment where students, researchers, and partners can work with real fish, real systems, and real-world challenges. By bridging the gap between academic research and industry practice, the center will strengthen Maine's aquaculture center and help ensure its workforce is ready to meet the demands of a rapidly evolving field. Next slide, please. While my role connects across many facets of our research, today I want to shine a light on the excellent work being done by our faculty team. Next slide. I won't spend much time on my own work today, but I do want to give a quick snapshot of the research areas I oversee: aquatic animal health, microbiology, and policy. In my role as the director of both the Aquaculture Research Institute and the Aquatic Animal Health Lab, I support a wide range of research and outreach efforts aimed at helping producers and policymakers make informed, science-based decisions. My focus includes disease management, biosecurity, and ecosystem health, key building blocks for a resilient aquaculture industry. I'm also especially proud to be part of launching our new innovation center. It's a major step forward in strengthening applied research and growing Maine's workforce. Now I'm pleased to turn things over to our faculty researchers to walk you through the exciting projects they're leading. First up is Sarah Turner.
Hi, everyone. As Debbie said, my name is Sarah Turner, and I'm a newly appointed Assistant Extension Professor and Aquatic Animal Health Specialist. I've been with the university for over 15 years now and manage the high containment wet lab facility at the Diagnostic and Research Laboratory. We host several undergraduate interns each summer throughout the year, as well as graduate students Amber Johnston obtaining her PhD looking at infectious salmon anemia virus affecting Atlantic salmon. And we just graduated our master's student, Daniel Delap, who's moving on towards a DVM PhD dual degree program. In addition to our students, we have technical staff like Bobby Harrington, who is an aquaculture systems specialist and keeps our wet lab facilities operational, and also is looking at water quality issues in recirculating aquaculture systems. In addition to our high containment wet lab facility, I also conduct research towards developing improved vaccines and explore how we can use sustainable biomaterials to help solve challenges impacting the aquaculture industry. I have extensive experience as a project coordinator for industry and grant contracted research in a wide variety of areas impacting aquatic animal health. Next slide, please. Disease is one of the most limiting factors for successful sustainable aquaculture, with outbreaks accounting for approximately 40% loss of production potential, which has been estimated at about $102 billion in losses. These pathogens can be bacterial, viral, parasitic, or fungal in nature, so a one-size-fits-all approach is not sufficient to tackling this problem. Because of this, outbreaks are managed through a variety of methods, such as use of probiotics, biosecurity, and best management practices, as well as providing high-quality nutrition. And you'll hear more about these areas from our team in a bit. In addition to these strategies mentioned, the aquaculture industry is dependent on vaccines and the development of new therapeutics to remain viable. Next slide, please. Our unique high containment aquatic animal health facility at UMaine's Cooperative Extension and Diagnostic and Research Laboratory helps us to address the challenges faced by these pathogens with the ability to conduct advanced research on new and emerging aquatic animal health issues proactively in a high containment setting. This allows for high level of government and industry collaboration and supports faculty and student research, as well as provides a pathway for product development. Performing in vivo safety, antibody response, challenge efficacy studies are the initial steps to licensing and commercialization for development of new vaccines or therapeutics, and ultimately towards solutions to mitigate disease. Our unique facility allows for this necessary R&D to support the industry. Next slide, please. In addition to our high containment suite, we also have standard laboratory spaces to perform cell culture and virology, microbiology, molecular testing, assay development, and we have access to on-site histotechnologists and veterinary pathologists. Next slide, please. For research programming, I'm currently digging a little deeper into how Maine's sustainable biopolymer resources can be a resource for the aquaculture industry. Biopolymers are polysaccharides produced from biological sources, such as carrageenan or alginates from seaweed, nanocellulose from wood fiber, chitin and chitosan from crustaceans, alginates from algae, and collagen and gelatin from fish sources. These biopolymers are biodegradable, sustainable, and biocompatible materials that are readily available in Maine and have shown recent significant improvement in areas such as increased efficacy of vaccines and drug delivery, tissue regeneration and wound healing, nutritional diet supplements, sustainable product packaging, the necessary backbone for bioplastics to replace traditional plastics, and also water filtration, PFAS mitigation, and effluent treatment. Next slide, please. One example of a project is a USDA-funded grant looking at using the most abundant biopolymer on Earth at the nanoscale, called nanocellulose or CNM. CNM is produced from wood fiber here at the University of Maine. It's sustainable, biodegradable, biocompatible, and highly tunable cellulose at the nanoscale. It's manufactured in industrial quantities at relatively low cost and is also very stable for production and storage. Overall, CNM meets rigid medical requirements and is an attractive and practical material with great potential to support finfish health. Next slide, please. Over the past 4 years, we've collaborated with UMaine's Biomedical Engineering Department and tuned CNM in 4 different ways as a vaccine system for Atlantic salmon. First, we looked at unmodified CNM, which showed promising results and demonstrated safety in Atlantic salmon parr. Next, we altered the CNM to be a stiff hydrogel. Then we chemically modified the CNM to be an easily injectable formulation. And finally, we settled on a hybrid that was both easily injectable and formed a hydrogel after injection. Next slide, please. We were able to test these formulations in our high containment facility where we acclimated the fish in our system for 14 days. We vaccinated them with our different formulations compared to a currently used vaccine found in industry. We let the fish hang out for about 50 days to generate an immune response, and then we performed sampling. Next slide, please. We then challenged our population of vaccinated fish with new fish that were infected with a common pathogen found in aquaculture, and we monitored the outbreak and then confirmed presence of disease. Next slide, please. While a little bit harsh, these small-scale studies allow us to identify which vaccine formulations significantly improved outcomes during a disease outbreak, as circled in blue, and which vaccines formulations did not improve outcomes, as circled in orange. Next slide, please. In addition, because of our standard laboratory facilities, we're also able to look at impacts of tissue at the microscopic level using histology, looking at changes in gene expression using molecular assays, and quantifying antibody response to our vaccine formulations using immune assays. Next slide, please. Results from this one project has we formed an interdisciplinary collaboration between ARI and the Biomedical Engineering Department. We've informed multiple industries and research areas on CNM potential as a vaccine component for aquaculture. We graduated 3 biomedical engineering master's students, and we have a patent filed while working towards commercialization with industry partners. Next slide, please. And while I provided just one example of how we use our wet lab high containment facilities here, I do want to highlight all the industry partners and grant funding resources that we've worked with in the past or are currently working with. And with that, I'd like to now introduce my colleague, Dr. Sarah Barker. and she will dive into a little bit more about vaccine development and disease mitigation. Thank you.
Right, just figuring out how to unmute myself. Hi, my name's Sarah Barker. Thanks, Sarah Turner. I joined UMaine last summer in July as an aquatic animal health specialist and assistant extension professor as well, with a joint appointment between Cooperative Extension and the Aquaculture Research Institute. I've worked in the aquaculture field for over 20 years now, with a focus on fish health, particularly vaccine research and R&D and host pathogen interactions. So my background kind of spans both academia and industry, where I previously worked for around 10 years prior to this position in an animal health biotech company focused on aquaculture vaccine R&D. So my interests are specifically around practical science-based solutions that improve fish health management on farms, with kind of two key areas being the development of disease prevention tools, so research into things like vaccines, and secondly, working with industry to develop new strategies to improve animal robustness during production, for example, like seawater transfer of salmon. And today I'm going to focus really on the salmon industry with what I'm going to discuss. So when it comes to fish health, the industry really lives by the motto that prevention is better than treatment. So making commendable and successful strides in disease control through the use of tools like biosecurity plans and best management practices, including things like careful site selection, lower stocking densities, parasite monitoring and removal, strict hygiene, fallowing, and area coordination of sites, disease screening, genetic selection, and vaccination programs. And when necessary, the use of medicinal and non-medicinal treatments, along with quick and planned responses to disease. And I should also highlight that new production methods and artificial intelligence are also helping expand these tools. However, changing environmental conditions and limited treatment and vaccine options make disease control more challenging. That's why it's important that we keep innovating, expanding our tools, and closely monitoring pathogen trends, both locally and globally. So the main goal of the research and education program that I'm developing in my new role is to support the salmonid industry's continued efforts to develop innovative strategies and tools that address current and emerging health issues. In turn, helping to support the industry's sustainability and the nation's food supply. So this will be taking a multi-pronged approach. First, working to better understand pathogen diversity, looking at what pathogen strains are out there and how they vary genetically. And this helps us to stay ahead of evolving disease threats and also design more effective vaccines and diagnostic tools specific to our region, as well as exploring alternative treatment options, such as alternatives to antibiotics for bacterial diseases, and to support disease mitigation through collaboration with stakeholders on risk management of environmental and husbandry practices, while engaging with stakeholders through education and knowledge transfer. And I just also wanted to reiterate that through the facility that Sarah Turner just highlighted, we can assist industry partners with drug and vaccine approval studies as well. So next, I'm just going to briefly highlight a project focused on bacterial diseases of salmonids that we're in the planning phases of, that will be integrating the kind of top 3 approaches of this research program that I just highlighted. So the first goal of the project is to understand the genetic diversity of key bacterial pathogens affecting salmon farming globally, but for which there's still a gap in high quality localized genomic data from the US. So we'll be addressing this by sourcing and collating bacteria isolates from the US and Canada. And through the use of molecular tools, we'll be zooming in on the whole genome of the bacteria to characterize the genetic variation, and doing this to kind of build a clearer picture of the bacteria affecting salmon farming in the US. This can then be shared in public repositories, and this will help support broader vaccine and diagnostic development efforts and foster broader scientific collaboration. And understanding this diversity is really essential for staying ahead of emerging threats and also to develop US strain-specific disease challenge models, to test new treatments and vaccines that are really relevant to our geographic location. And it's also essential information needed to design vaccines that target the right pathogen strain and improve diagnostics. So why is it really important to understand pathogen diversity when it comes to vaccine development? So just like in humans, pets, and terrestrial farmed animals, vaccines are really key tools in aquaculture to protect fish from disease. So some vaccines in the industry work really well, but when it comes to making effective new ones, it kind of starts with understanding the diversity of the pathogens which are causing disease in the field. So this diagram shows a very, very, very basic vaccine development process. So it starts with target identification. So which pathogens do we need to protect the fish against, which species and strains? Then it goes into antigen design. So what part of the pathogen will trigger a protective response in the fish? And then platform selection. So there's different ways that you can present that antigen from the pathogen to deliver that protection in the most effective way. And then obviously testing. So is the vaccine safe and effective in the fish? And then if these are all successful, it goes on to scale up of production. So from small lab batches in research to large manufacturing volumes in the hundreds of liters. And then a whole host of studies for regulatory approval with the final manufacturing and distribution to get that vaccine out into the field. So understanding this pathogen diversity is really especially critical in these early steps on the right-hand side of the wheel. And it helps, as I said, identify which species and strains are causing disease, what antigens to include in those vaccines, and how the vaccine should be designed to offer the best protection, especially as certain diseases aren't always caused by just one type of pathogen species. Sometimes different species or strains are responsible. And for instance, if you have a new strain that appears, current vaccines might no longer work, requiring updates in the formulation to keep fish protected. To be able to use this information and support research into new vaccines, we're also setting up a dedicated lab at the Diagnostic Research Lab, which will be focused on developing and formulating aquatic animal vaccines at lab scale. So this will be using recombinant technology to design vaccine antigens and platforms while having the ability to test their efficacy and safety in our high containment facility on site that Sarah Turner highlighted previously. So we're hoping that this initiative will strengthen our partnerships with industry to support the creation of vaccines and diagnostics specifically tailored for the US with our industry partners. Beyond vaccines, our second goal is to explore potential alternatives to traditional antibiotics. So we have a current collaboration with Medical University of South Carolina to evaluate a natural, sustainable, plant-based antimicrobial alternative that they've developed. And we'll be testing this against bacterial isolates of importance to the salmonid industry. And we're hoping that this work will really support the longer-term goal of identifying effective, environmentally friendly treatments, especially in light of increasing concern around antimicrobial resistance. But we don't want this work to kind of exist in a vacuum. So that's why our third goal is centered around engagement with a dedicated stakeholder workshop plan to bring together US salmon producers and various colleagues from ARI, Cooperative Extension, and Sea Grant, as well as the biotech industry. And this would really be to kind of focus in on three key areas. So reviewing current diagnostic and management tools, summarizing the project findings and where the knowledge gaps still exist, and to really have an open and collaborative discussion on prevention strategies moving forward. So this is really a critical step in making sure that our research stays grounded in the realities of production and helps drive solutions that are timely, targeted, and scalable. I'd also like to very briefly highlight a couple of other projects in the pipeline. So first, we have a very exciting partnership with the USD ARS to look at a major challenge in salmon farming, the salmon louse. Sea lice are parasites that feed on fish skin, mucus, and blood, causing problems for open sea salmon farming worldwide. And the industry really needs more tools. So this collaboration aims to develop improved prevention and treatment strategies, And the last one is a collaborative project within industry that we've been pulling a proposal together, which we're seeking funding for, which is really to support hatcheries for salmon to assess water-related risks to fish health, as water quality is really fundamental to the health and success of hatchery operations. And so that's really a quick whistle-stop tour. And now I'm going to hand over to my amazing colleague, Matt Hawkyard.
Thank you, Sarah. So I'm Matt Hawkyard. I'm an assistant extension professor and finfish nutritional specialist here at the Aquaculture Research Institute. My research areas are in early life nutrition of finfish, feed technology, and fish nutrition. I have many projects going on, but really what I want to talk to you today is about focusing on the development of improved starter diets for marine finfish, early stage finfish larvae. Oh, next slide, please. So the challenge with these, with early life history of finfish is that these are pretty much among the smallest vertebrates on Earth. I'm here, I show a typical early newly hatched salmon, 11. You see that there's kind of the belly is protruding. That's actually the yolk sac. So this fish is still receiving a lot of its nutrition from the yolk, but as soon as that's gone, it's going to be reliant on external nutrition. On the bottom, I'm showing a couple California yellowtail larvae. The bottom most fish is actually like a newly hatched, it's about 7 millimeters long. So these are all to scale with that penny. So you can imagine they're very small, and that has some challenges. But next slide, please. It's not just about being small, they're also very poorly developed. And so we're trying to feed these animals that in many cases, they don't even have all their organs developed. They might not have a stomach yet. Their digestive system is just a basic tube at this point, at those earliest points. And then those systems begin to develop over the period I'm talking about. But that means that they have limited ability to digest food, and also their fins aren't developed, so they're not going to be able to obtain food, swim around, and actively find prey or inert feed particles nearly as well. Next slide, please. So there's consequences to getting this right or wrong. If we have good early nutrition, thank you, that will lead to better, more rapid growth. Can lead to better, more rapid growth and setting up trajectories that last long beyond these early stages. That also can lead to better resistance to stressors and disease, a term we call robustness often. And also, getting early nutrition is important for getting the early development correct. And so we want to make sure these animals are normally developed so that, that leads to better animal welfare and also consumer preference for as these things mature. Okay, next slide, please. With marine finfish such as like California yellowtail, they actually have these that we call them matricial larvae, but that means very poorly developed larvae that can be reliant on these live feeds. So this is showing a feeding regime for California yellowtail where they start them on rotifers, transition them onto artemia, and then eventually get them onto an inert particle more similar to what we feed juveniles, although these are very small particles. That has its own challenge. But you can imagine we're now growing organisms to grow organisms. There's risk and labor associated with growing these live feeds, and it represents a lot of cost for these commercial hatcheries. Next slide, please. So on the left, I'm showing a starter diet, which I'm going to refer to as micro diets. It's about a 0.3 millimeter micro diet on the left. And on the right is a juvenile pellet. So you can kind of get an idea of the scale, the difference between what you might be more familiar with. The 3 mil pellets, something that might look a little bit like cat food if you haven't fed fish before. But these small particles look like dust, and you're just sprinkling them in the water. And there's a lot of technical challenges that go along with making those diets perform adequately. Next slide, please. So the characteristics that when we're formulating and producing or working on from a feed technology standpoint, developing these micro diets, we have to think about lots of things like, will the fish eat it? So acceptability and palatability. Do they taste good? Do they have the right kind of mouth feel? Do they sink too fast for the particles for the fish to have access to them, or do they stay on the surface? So sink rate is important. They have to be formulated to have the right nutrients to meet the nutritional needs of the animals. Those nutrients have to be in a form that's digestible. For the ingredients have to be digestible, and also if there's any sort of encapsulation done, that has to be digestible. And these are poorly developed animals with not very good digestive systems. So, there's that's particularly difficult. The nutrient retention is important. So this feed actually has to pass through the water, meaning that things that have a high affinity for water will leach out really rapidly, which is something we are constantly trying to solve, and it's very challenging. And finally, we have to do all of that in the context of keeping them cost effective and competing with the cost of live feeds and also the cost of kind of current commercial technologies. Next slide, please. So what I really wanted to focus on today is our open formula concept that we've been developing. So we have an open formula diet that we make for marine finfish larvae. And what I mean by that is this, you may have heard the term open source, meaning, publicly available, transparent. In this case, we're talking about an open formula, which is the same thing. This is the recipe of our open formula on the right. We share this both publicly with other researchers and with commercial feed companies and hatcheries, so that for two main kind of points from a research standpoint, having an open formula like this that researchers can take advantage of will help kind of harmonize our research across the broader research landscape. And we're really kind of focused on North America here. At some point, if enough people adopt this can actually be used as a reference diet. In other words, a control diet that we constantly put in our studies, so that we don't have to use a commercial diet for those references, which commercial diets are, the opposite. They're closed formula. We don't really know what's in them, and they can change over time. Yep, next slide, please. So we actually produce our own open formula micro-extruded marumerized diets. These are micro diets in-house. This is the micro extruder on the left, and you can see at this point we're making the noodles. Then in the middle, that's actually the spherinizer that takes those noodles and spins them on this rotating disc, and that creates little kind of beads. And then on the right, we then take those beads and we put them into the fluidized bed dryer that dries them down to improve the shelf life. There's a few more steps. We have to, mix the ingredients, weigh them in, all that, package them. But this kind of gives you a general idea. Next slide, please. So the cool thing is we have now tested our open formula reference diet with California yellowtail with a few different trials, actually. This one was pretty recent and pretty exciting in that our diet, which is the green bars there in the middle, the OFRD, performed as well as the leading North American diets that are typically used in North America. That's Otahime in purple, and Alltech is another common one. Our commercial partner Ziegler are developing a diet, so that's the orange one. We did have a company from Norway do quite well with their diet, that blue one, so that's great. That means we have room for improvement here in our North American diets and our open formula. We can start making some tweaks to to that open formula to then improve the growth performance of fish in the future. But in general, what we wanted to do was just perform as well as the kind of standard commercial diets on the market, so that we can start using this as a reference diet here in North America. Next slide, please. So what we're doing now that we've tested this, we're trying to test it with a greater number of species and really share out this formulation, this open formula concept with a number of groups. So one thing we do is we're offering 1 kilogram of these open formula MEM particles to any group that wants to test it with a new species that has commercial potential, particularly here in North America is where we're focused with that program. And but we do have some early adopters and users and commercial companies too that are using our open formula at least as a reference, and in looking at, they can then compare it to their own internal formulations and take kind of parts of it that they like that might work for them and adopt it. So we have a couple hatcheries that are testing it at present, and then we have a couple of feed companies as well as some research partners, both in the US and abroad. Next slide, please. So finally, the goal is not to just let this sit. It's to use this open formula reference diet concept to then improve feed technologies in the future. So one thing we're working on at present is applying protective coatings to the outside of these commercial type diets. So on the left, you can see a SEM picture of a non-coated diet. On the right, the bottom picture is a scanning electron microscope of a coated particle, and that's really to improve the nutrient delivery as these things pass through the water. These kind of have a capsule on the outside. It has to be digestible and all those things. But anyways, this kind of gives you an example of that. One of the ways that we're improving technologies. So with that, I'll hand it over to my colleague, Dr. Michael Haptasian, as he talks more about some finfish nutrition work. Thank you.
Thanks, Matt. My name is Michael Haptasian, Assistant Extension Professor and the Fish Nutrition Specialist with Aquaculture Research Institute. I conduct research in fish nutrition and nutrigenomics. Also, we evaluate different alternative protein sources, carbohydrate, and oil sources in finfish diets. We investigate dietary supplements and specific nutrient requirements for finfish. Our research is targeted on aquaculture farmers and feed production industries in the state of Maine and in the United States. I have established collaborations within UMaine, US universities, industry stakeholders, also international collaborators. I also train students, both undergrad and graduates, also postdoc. Next slide, please. As you know, aquaculture is one of the fastest growing food production industry globally. As you see the figure, it's increasing very fast up to, you can see the projection from early up to 2032. For example, in 2022, world aquaculture production reached its new record, about 130 million tons, which is increased by 6.6% from 2020. Next slide, please. However, aquaculture is also facing challenges, especially from feed costs, which covers 50 to 60% of the production costs. Mainly, these challenges are from the traditional ingredients, including fish meal and fish oil. As you see in the figure, world fish meal production is declining and the cost is increasing. The other challenge is disease outbreak. It's already mentioned by my colleagues from the aquatic health team. Next slide, please. To evaluate those alternative ingredients, we have received many projects, both industry contract and research grants, including insect meals, black soldier fly meal, and the mealworm. These are industry grants from Insect and Stratium. Single cell protein from low value wood biomass. This is grant. We are going to evaluate single cell protein in the diet of Atlantic salmon. Novel plant protein, fermented kelp in the diet of Atlantic salmon. This is also a grant. Seafood byproducts. We evaluated clam meal in the diet of Florida pompano. This grant. Carbohydrate sources, oil blends, also specific nutrient requirements in different finfish studies. Advantage of those alternative protein sources, they are novel protein sources. They contain from 40 to 70% of crude protein based on their processing or production process. They have favorable amino acids and fatty acids profile. They are also functional and cost-effective ingredients. They also use limited land and also water. Our overall goal is sustainable feed development for finfish aquaculture in Maine and US, mainly focusing on cost-effective, nutritionally healthy, and also environment-friendly aquafeed in Maine and the United States. Next slide, please. To start with our research, the first thing is feed formulation. We formulate diets based on the nutritional profile of the test ingredient, then followed by feed production. We use the USDA facility in Bozeman, Montana. Facility is well-established feed production line, including from fish weighing, mixing, pulverizing, extruding, drying, cooling, oil coating, also bagging. Next slide, please. After feed production is fish rearing, we obtain fish juveniles and stock them in our systems. Already Sarah Turner explained it. Then we acclimatize them with the system for 2 weeks, then assign them to diet 3 or 4 replicates of each diet. Then we will have daily feeding 3 times per day for 12 weeks. This is for growth study. And we maintain the water quality. Challenge experiment is optional. If we are developing functional feed, we can challenge fish after the end of the study. Next is sample collection. At the end of the study, we collect different samples, starting from fish weighing, collecting different samples, including whole body, plasma samples, organs like intestine, liver, head, kidney, and others. Next slide, please. Research approach, we do comprehensive nutritional research evaluation to evaluate the alternative ingredients, starting with digestibility study to see the digestibility or palatability of the test ingredient. This is short study, 2 to 3 weeks of study. Then growth study, as I mentioned, it takes 12 weeks or 3 months. Then we measure growth performance, nutrient utilization, nutrient profile, biomarkers related to fish health. At the molecular level, we do gene expression, gut microbiome. Bacterial challenge also as an option to develop functional feed. Next slide, please. Our research findings, I selected 2 studies. So this study is mealworm evaluation in Atlantic salmon diets funded by WINESECT France. We evaluated the effect of mealworm on the growth and the health of Atlantic salmon. We formulated 4 diets: control diet, which is 100% fish meal, 50% replacement of fish meal with defatted mealworm, and complete replacement of defatted mealworm. Also, the 4th diet is 50% replacement of fish meal with whole mealworm. As you see, the growth performance, we weighed fish at the beginning, midpoint, also at the end of the study. We didn't find significant difference, which is the test ingredient performed similar to fish meal. Next slide, please. At the molecular level or gene expression, mealworm, especially the defatted one, upregulated the gene expression of immune-related genes in Atlantic salmon, which indicates the immune function of this ingredient or mealworm. On the right side of the slide, gut microbiome, Pseudomonas was the dominant genus which is involved in hydrolytic enzymes production, which could help the digestion capacity of fish. Next slide, please. The second study is defatted black soldier fly meal evaluation in Atlantic salmon diet. We did comprehensive nutritional evaluation. We checked fish growth performance and health parameters. First, we formulated 7 diets, a control diet, and 6 test diets. This is factorial design. We used 3 levels of defatted black soldier fly meal and 2 levels of soy. As you see, the body weight, we weighed fish at the initial, midpoint, and final. At the end of the 12-week study, we found some difference. The high replacement of fish meal with defatted black soldier fly meal a little bit reduced the growth of fish. At the molecular level, which is related to health, on the right side, we checked immune-related genus and the defatted black soldier fly meal. At 15% of the diet, upregulated this genus, which indicates the immune function of the defatted black soldier fly meal for this fish. Next slide, please. Summary and conclusion, overall, mealworm meal could replace fish meal in Atlantic salmon diets and improve fish health. This is already published in Aquaculture Nutrition last year. Defatted black soldier fly meal up to 15%. This is limited now of the diet. could replace fish meal without compromising Atlantic salmon growth and health. This also published in Aquaculture. Our overall conclusion is these findings would support the sustainable feed development, specifically nutritionally balanced, cost-effective, and health aquafeeds for finfish in Maine and the United States. Thank you.
Thank you. So we do have some questions. And I'm going to ask the question, and then if any of the panelists feel like they can answer the question, please do. I'm going to take the first question, and that is, how do regulatory policies impact innovation in finfish aquaculture? I would say that in Maine, most all of our resource agencies have some type of regulatory requirement. Like for importing a new species, we have to bring them into quarantine, so we have to do it through the Department of Marine Resource regulations. If we're developing new vaccines, novel vaccines, we're under the USDA APHIS and Center for Veterinary Medicine, and in that case, we just have to prove safety. These are all things that we can do here at UMaine. And with food innovation, and that tends to be, nutrition tends to be one of our highest priorities. That's a matter of working with the industry and making sure that the food is recognized by the FDA as a formulation that has to have everything that's approved by the FDA to put in food feed. Maybe Michael can answer that a little bit more clearly.
Thank you, Debbie. So the process for FDA approval starts with research findings, and the research findings helps or can be used as evidence of the nutritional profile, as well as the application of the test ingredient for animals. For example, we tested clam meal in Florida Pompano in 2021. And Sea-Watch International, they applied for FDA approval of their test ingredient. And this company, they used our research findings, which published in scientific reports, Nature. And that publication helped them as evidence to get approval, which is the initial approval for FDA as feed ingredient or alternative feed ingredient for Florida Pompano for finfish. Thank you.
Okay, our second question is, and this is open to our panelists, what opportunities exist for students to engage in applied research projects at ARI?
I don't mind jumping in on that one. I think importantly, ARI employs a lot of students in the research that we do. And it's a big part of our mission is, teaching and workforce development come right along with this. So we have both at all levels. We have undergraduate students that come in and through the University of Maine particularly that are doing capstone projects and other internship-based programs. And they're directly involved in kind of the day-to-day husbandry and research, activities. We also have externals from non-UMaine students come in through a research experience for undergrad program where, we'll work with those, particularly over the summer, students over the summer period to develop projects and work on kind of sustainable aquaculture questions. And we learn from them as well. We also have graduate students, and most of our projects employ at least one graduate student, I'd say, to take the lead on a lot of the research activities. And then we have continued education that goes on as well, and that kind of goes into that workforce development point. So a lot of teaching and training that goes along with, professionals that want to learn more to further develop their career in aquaculture.
Yes, I would say that our workforce development programs, are either the traditional undergraduate, graduate, and then lifelong learners through programs that we're actually developing with cooperative extension. So the third question is, what events and programming does ARI coordinate or host to promote knowledge sharing with the industry? We do that by actually attending conferences and presenting our work at conferences. Many of us deal directly with industry stakeholders, and so we report our work to our industry stakeholders. We very much appreciate being part of MAIC's Research Education and Summit. I forget what the actual name of it is called, but it is the summit that occurs for Maine Aquaculture Research every other year. We participate in NACE, amongst other things. We host a student webinar series to just keep up with everything that we have that we're currently doing.
I think I can also add, Debbie, on that as well, because we highlighted a bit in the presentation, but just where appropriate as well, when we've got certain projects, we might host workshops to kind of give those results back and have discussions with industry on them.
More and more of the recently funded grants that we've been awarded do require the extension component of actually hosting a workshop. So our big, we call it Trees to Feed, our sustainable wood fuel and fish feed actually has a very strong component of extension and workforce development in it as well. So the last question is, okay, there's two more. How does ARI and applied the Aquatic Health Lab collaborate with industry to address applied research needs? How can industry partners reach out to you if they have an emerging need? That is how we've engaged with, over 20 international companies. They have we actually don't do a lot of advertising except what is on our website. We have a portal on our website that allows people to ask questions. That's usually our first introduction, or just by someone telling them the work that we do. At the university, we're very fortunate to have the Department of Industrial Cooperation, which helps with all of our material transfer agreements, all of our non-disclosures, setting up protocols and deliverables. It is, as a contract research organization, it is a cost per service, but for Maine-based companies, we actually give a prorated cost for them to be, for Maine industry. Last question is, what future trends do you foresee in Maine's aquaculture industry that may require further research and innovation? I've done a lot of talking, so would someone else like to answer that?
I can jump in for a second. So speaking more on like the health side, but also on production, I think it's just kind of adoption of new production methods. So whether it be recirculating aquaculture systems or growing post smolt, so bigger smolts that go out to sea when we talk about finfish with salmon, and how those might increase robustness of fish or reduce disease threats. But also then how do you input all those best management practices and adapt those to those new production methods as well. And just kind of the integration of artificial intelligence that we're seeing globally, like how that might play a role in Maine's industry as well. Yeah, I don't know if anybody else wants to jump in.
Again, I can say that through some of the partnerships that we have, like with the Institute of Marine Environmental Technology, we often do ask the industry what their top priorities are. The MAIC and ARI participate in a survey, and those are the innovations that we see. And again, it's more resilience to a changing climate. Are we allowed to say that? And alternative feed, improved feed development, improved health, development of new species, all of those, even in finfish as well.
I think the other thing I'll add is, this is not a kind of a rocky road, but potentially the movement of the development of land-based aquaculture systems for finfish. And so, we refer to them as recirculating aquaculture systems, or RAS. And as those facilities, if we start seeing those commercial success there, we'll be seeing a lot more development in that area. And that means for us to facilitate that, feed technologies that are going to work specifically well in those systems, because there's a lot of special considerations that go along with these land-based systems. And then similarly, I'm sure from a disease standpoint, you have some kind of specific considerations and methodologies with respect to those. So I think that's kind of one of the things that we see, and not just like moving from one system to the other, that means from sea to land, but even hybridization. We see that as like a current model where companies are just extending the earlier life stages longer and longer on the land-based systems, so that they can have a little bit more, say, climate resiliency, and also it kind of opens up their production sea systems with a little more space. So kind of both will be developing, and then also in combination in this kind of hybrid system. But all that comes with a lot of kind of special research needed to kind of meet those, facilitate that kind of expansion.
The other thing I would add is functional feed development, which is very important, and it's one of the recent research approaches for tackling disease outbreak. So I would say functional feed development also is very important.
Thank you. That's the last of the questions.
Awesome. Thank you all so much for a fantastic presentation. I learned a lot. Keep an eye out for our next month's webinar. It's going to be on June 20th. And thank you all for tuning in.